So chapter 31 is kind of a long chapter, and we'll talk about plant structure and growth really just a little bit. But humans have always relied on plants for I just put those two words together. Food, fuel, shelter, clothing, and countless other necessities and niceties of life, things that make life nice. About 11,000 years ago, a major shift occurred, and we actually began to cultivate. So properly sow rather than just gather crops. Cultivation was soon followed by domestication. So we had those genetic changes in crop species resulting from selection by humans with desirable traits. So the domestication of plants is one of the most significant events in the development of human civilization. Angiosperms, the flowering plants, make up 90% of the plant kingdom at this point. So the whole showy flower business is, it's working. So it's kind of selected for. So there's an angiosperm, there's more angiosperm, there'll be lots of angiosperms. So we're going to look at plant structure and function today and plant growth. So we'll do this here, but our essential question will be, what is plant structure? And then we also want to look just a little bit at how do plants grow. So those are our two essential questions. And we'll spend a lot of time in plant structure. So to illustrate the long-standing relationship between humans and pretty flower angiosperms, let's start with the history of a single domesticated crop of wheat. Wheat accounts for about 20% of all the calories consumed worldwide, and in the U.S. it's one of the most valuable cash crops, with American farmers exporting about 30 million tons of it each year. Before about 10,000 years ago, all the wheat found in human settlements was wild, but about 10,000 years ago, wild wheat began to be replaced by domesticated varieties. Today, virtually all of the wheat cultivated around the world is one of the domesticated variety. All right, so just so we kind of see what happens is we've had a shift. This is also sort of evolution, although its mechanism is artificial selection. We still see a change in that population over time. So some other kind of thinking, genetic analysis also was used to pinpoint the time and place of domestication, and such a genetic analysis indicates that corn was the first domesticated about 9,000 years ago in southern Mexico. And botanists have been able to piece together an overall map and timeline of crop domestication throughout the world. So a little like historical fact on that stuff, where these guys are coming from, which is really interesting. All right, so that kind of brings us, we got some good history out of the way with that, but we're going to talk about our two major groups of angiosperms. We've got monocots and eudicots, which I learned as dicots back in the day. So if you just hear me call them dicots, I'm still talking eudicots. All right, so monocots and eudicots differ in a number of ways, and this is actually important, so it's worth noting. But they differ in the number of cotyledons. These are little seed leaves. So if you have a seed and it grows, there will either be one little leaf or there will be two. Okay? So those are seed leaves. They also differ in their pattern of leaf veination. So they're leaf veins, the arrangements of stem vascular tissue, number of flower parts, and what the root structure looks like. So these are some things we're going to talk about, and we're going to add to our mini project. So monocots, like wheat and corn, so these are two good examples worth having. They have one cotyledon. Notice the name mono. We should know that as one, and cot for cotyledon. So it is a one cotyledon. Look at that. They do have parallel leaf veination. So a lot of these guys, you could think like a grass type thing, and wheat's that. The leaf veins are going to sort of go this way. And you'll know what I'm talking about when you see it. We're going to have scattered vascular bundles. So if I have a plant and here's like maybe some leaves or whatever, if I cut it at the stem, what you'll see is these little bundles that are kind of scattered throughout. It's going to look like that. Flower parts would be in multiples of 3. So if we do this, that'll work. Terrible, but it looks like 6. And we'll put a stem on it. Yay! It's also going to have fibrous roots. So there's just some goofy drawings just to kind of show you what we're talking about. And roots would be like, here's the thing about the, but underneath the soil, it's just going to be like roots that are kind of, they just go out like that. Eudicots, we'll have much better pictures, I promise, have two cotyledons. This is why I learned them as dicots, because we have di or two and cotyledons or cots. So they're dicots. They're going to have branched leaf venation. So honestly, this is what happens whenever you guys draw a leaf and you go like, oh, look at this cute little leaf. That's that leaf venation that you're doing. There's going to be a ring of vascular bundles. So again, when we do a cross section of the stem or different places, we're going to see that there's vascular bundles that are going to kind of be in a ring around, kind of like that. Flower parts will be in 4s or 5s. There. There you go. You get, that's my best. So we got 5 little petals around a center point. There you go. And these are also going to have a taproot system. So that means here's our little plant above ground. Underneath the ground, it's just going to have like a long root that's kind of really goes far down, and then it'll have like little tiny root hairs that come off of it. Maybe a few other little bits, but the main thing is that there'll be a longer root that taps down into the water resources below. All right, so here's sort of what we look like. We'll have to look at cotyledons a little bit differently, but these are the seed leaves. So 1 will be in monocots and 2 in dicots. You'll see the leaf veins are different. So in monocots, notice they are parallel. So you can see those things running parallel to each other. And now that you look at it, you go, oh, that makes sense. I've seen those. Eudicots, you can see the branched leaf veins. Stems, notice there's kind of a scattered bundle. They're just kind of in there. Whereas in our eudicots, they form this kind of ring structure. If we look for monocots and their flowers, it's usually in multiples of 3. So you can see that here, 1, 2, 3, 4, 5, 6. So that covers it. In our eudicots, it's usually 4 or 5. So you can see 1, 2, 3, 4, and 5. And even out here, it's 1, 2, 3, 4, and 5. Okay, so we can see that there. The roots for monocots are fibrous, so they look sort of just kind of like a net kind of going out. But for dicots, it's usually a taproot, so it's going to go straight down. Okay? So those are some important differences. Plant organs consist of several types of tissue that carry out particular functions. So we'll talk about some of those organs. We got to know piece by piece here. So plants use a root system to anchor the plant to the soil, absorb and transport water and minerals up, and also to store some carbohydrates. So those are our sugars and stuff like that. We have root hairs, and these are tiny finger-like projections off the roots that enormously increase the surface area for absorption. So we'll look at that example here in just a second, but it's good to know these parts. Okay, that's our roots. This is our shoots. So, basic idea. Here, I'm going to make it like a flower. It's the best I can do. So, roots. These are shoots. The roots go down, the shoots shoot up. So, they consist of stems and leaves. And so, stem, leaf, and structures for reproduction, like a big ugly flower. Hey, it works. That's my doodle. And if you want better doodles, you doodle them yourself. All right, the stem also has nodes, and these are points at which the leaves are attached. So if here's a little stem, oh no, it's not my best. There, okay. So where the leaves are attached. So this point right here is a node, and this is a node, okay. The internodes, so inter actually means between. Oops. Move my whole thing over. Hold on. All right, but inter means between. So it's the space in between those two nodes. That's an internode space. The leaves are the main photosynthetic organs in most plants, And although green stems also perform photosynthesis, so we have it there, but these guys are mostly trying to catch the sun. All right. So we'll get to talk about some cool modifications a little later on. So just to take a look, we've got a root system, and we've got maybe a taproot here, so this would be one of our dicots. But you can see some root hairs, and these guys are increasing the absorption. So really, the root system is just roots and root hairs. I'll circle the big things to know here. All right. The shoot system, we have nodes, and so there's a couple of nodes here. Our internode is the space in between. We also have a leaf, and there'll be a blade and kind of a petiole to it, but we need to know the leaf, and we also have to know really the stem part. The other part we better know is the flower. Okay, I'll circle the node part too, but those are the big things there. The big things worth noting. All right, this is a good spot. We're at 11 minutes. Take a break, pause, and then come right back. So plants typically have two kinds of buds. We've got terminal buds, which think terminal is really like it terminates, it ends. So the terminal is kind of the end. So it's the apex, or really the top. Okay, with developing leaves and a compact series of nodes and internodes. So really it just means that we're going to have a lot of little nodes and internodes kind of close together towards the end. That's all it's saying. Axillary buds are found at the angles formed by the leaf and the stem. So we'll take a look at those in a second. But in many plants, the terminal bud produces hormones that inhibit growth of any kind of axillary buds. And this is what we call apical dominance, like the apex or the top is dominant in actually making sure that it looks the way it's supposed to look. Move on here. All right, maybe we don't have that one, but we go back to this picture. This is the terminal bud up top, and notice that our little internode space is pretty close together. So our nodes are closer together. And any axillary buds would be kind of in between where those two meet at. But sometimes our terminal bud will say, nope, 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 we're not going to have any growth in there because we want general growth to be up. Okay, just kind of depends on the plant, what the plant's trying to do. So modification of plant parts are adaptations for various functions, including food and water storage, asexual reproduction, protection, climbing, and photosynthesis. So this will be really helpful for kind of we can think about this from the videos that we just watched. So unusually large taproots that store food in the form of carbohydrates like starch are found in carrots, turnips, sugar beets, and sweet potatoes. So these are all those things where maybe you have stuff above ground, but then you've got kind of a large taproot. So definitely a carrot is a good example to think of here. Okay. But also turnips and beets and sweet potatoes do sort of the same thing. All right, so there you go. A little more of that. Stems can be modified in a couple of ways. So we have stolons, which are for asexual reproduction. So they're also called runners. If you've seen the common term of runners, that really is it. We'll take a look at some pictures of those. Tubers are storage and reproduction, so that's part of a potato. A potato is a tuber, and a rhizome is for storage and asexual reproduction. So let's look at some examples. But here's a stolon or a runner. So like a strawberry will have a little piece come off and it will end up somewhere else, which it will replant and then have another one come up. So it can actually sort of create another plant by just sending out some part of the stem. A rhizome and an iris plant actually store some things here as well. But we also find for potatoes, there's the tuber, which helps to store things, but it is a modified stem, which is different than the sweet potato. Just to note. These are some cool things. Leaves can be modified in a way also, so climbing like a tendril, that's something we definitely looked at in the video. Whenever you've got a leaf coming up and kind of wrapping around something and moving up and wrapping around and moving up, wrapping around and pulling itself up. Those are those tendrils. Okay. There'll also be protection like a cactus spine, but cactus spines are also modified so that way those things don't take in so much sunlight, because they don't have a problem with getting sunlight. They do have a problem getting water, so it actually limits photosynthesis because of water. So that's another thing, but it also is for protection. So here's a tendril. Also these cactus spines, which are definitely for protection. I know I've had a cactus, I swear to God, it reached out and bit me. It came at me. I don't know how, but it did. So the organs of plants contain some tissues, which are a group of cells that perform a specialized function, similar to human tissues. Think about, you have muscle tissue, you have think cardiovascular tissue. I mean, you've got tissues for different things, and they perform different functions. Same thing with plants. The two that we're kind of majorly concerned with are xylem and phloem. So xylem is our water conducting cells that convey water and dissolve minerals upwards. So xylem is the up. That's bringing the water from the roots and minerals from the roots upwards. So xylem is up. Phloem is tissue that contains cells that transport sugars and other organic nutrients from leaves or storage tissues to other parts of the plant. So xylem is up and phloem is really out. It can be maybe down, but it kind of flows out. It's got the sugars and it's flowing out. Okay. So where those nutrients are needed. All right, a lot of things to look at on this one, but the big part of this is just to see what those little bundles look like. So we do have some vascular tissue, and this is really, that's our kind of xylem and phloem. I'll call it X and P. That's our xylem and phloem. Notice they're colored purple in these little veins. But you can see the difference also between eudicots and monocots, is that in that dicot, they're in this kind of ring structure. You can see it here. They're around the kind of outside, the exterior there, and they're more bundled together. Whereas in a monocot stem, they're scattered all the way throughout. In a dicot, if we're looking at the roots, because it's not, I promise you it is not well done here, but these down here are roots. So this is root tissue. In a dicot, you'll find the phloem and the xylem, you'll find those in the middle, just concentrated there in the middle. Whereas in a monocot, it's going to be in the middle but larger. It takes up a larger space. So this is another way that they kind of look different. All right. We'll keep it moving here. There are a couple of things. I'm really just going to give you the basic definitions of these. I don't want you to harp on it too much, so we'll move quickly past this. But each plant organ, so root, stem, and leaf, has three types of tissue. There is dermal, and that's a protective outer covering. There's vascular, which supports and long-distance transport, so that's really our xylem and our phloem. And there's this stuff called ground tissue, which is the bulk of the plant. It does photosynthesis, storage, kind of support. So it just kind of, I don't know, best ways, it like keeps it grounded, it keeps it kind of plugged in and doing what it needs to do. So that's really the bulk of the plant. Much more than that, I'm not super worried about. Really, we have dermal tissues, and there'll be epidermis and cuticle. Vascular tissue, I'm supposed to take this out, but this is xylem and phloem, so you get the idea. Just do a brief kind of discussion, and you can see in the stem, one more time, we've got our nice ring structure in dicots and scattered in monocots. Okay, a little different in the roots again too. So in the center for those dicots, kind of a larger, maybe a little more spread out for the monocots. Ground tissue have this stuff called pith and cortex, which we won't get into too much, but it also has some mesophyll, which is helpful for photosynthesis and that kind of thing. Something that is important is that in a leaf, the epidermis is interrupted by tiny pores called the stomata. This is necessary to know. So the stomata actually will be part of exchange of CO2 and O2. So this helps kind of our oxygen get into the leaf so we can have photosynthesis happen. It's flanked by two guard cells, which means that we have an opening, and on the outside are these two guard cells, sort of like that. And so these guys regulate and can open and close the stomata as necessary. Okay, so basically it's not the best picture, but right here. So we've got our little guard cells, and that's really all they look like, is just two, I don't know, it didn't look right, but something sort of like that. And here in the middle is our opening. So our actual stomata. Most plant cells have three structures that distinguish them from animal cells. So just a good review of plants versus animal cells, because I know how quickly we want to forget these things. Do you remember they have chloroplast? That's for photosynthesis. They have a central vacuole that helps to keep the fluid kind of the way it's supposed to be, and it keeps them keeps the turgor, or it keeps them turgid. Turgid is the adjective for that, and so that gives them a certain firmness, so they're not all wilty. All right, the protective cell wall that's made of cellulose. So chloroplast, central vacuole, and cell wall are the major ones there. Some plant cell walls have two layers, a primary cell wall and a secondary one. Not really worried about that, but the big thing that I want you to remember is the plasmodesmata. So because we have this protective cell wall, right, around these cells, here's some nuclei for you. Because they have that protective cell wall, they have to have breaks within the cell wall to be able to communicate to each other and to kind of send signals and nutrients back and forth. So those holes that are poked within the cell wall are called plasmodesmata. Okay, they're really important. All right, here's a much better picture of those plasmodesmata, and then you can see the chloroplast and the cell wall and that central vacuole that separate them. Okay. All right, plant growth. We're going to do this one fairly quickly, but you can take a break here if you need to. So pause the video, go back, chunk your notes, write some questions out to the left side, and then maybe like start a summary on some of those pages, and then come back. All right, so most animals are characterized by determinate growth, which at a certain age you stop growing. I stopped growing. This is as tall as I'm going to be. My mind may think that I'm taller, but my body says, nope, you this tall, lady. So that's determinate growth. Our size is determined. Most plants have what we call indeterminate growth. They grow throughout their entire life. There's not technically any kind of a stopping point. Plants are categorized based on their length of life cycle. So we have annuals, and it takes them 1 year to complete their life cycle. Biennials complete their life cycle in 2 years. And perennials can live for many years. So we got annuals, biennials, and perennials, which you actually hear those if you go look for plants, so it's worth noting. Oh, I forgot what those were. It's like baby's breath, but I don't remember which one it is, so that's okay. So plant growth occurs in specialized tissues called meristems, and these consist of undifferentiated cells that divide when conditions permit. So undifferentiated sort of means they're stem cells. They don't know what job they're going to do yet. They can do just about anything. They're baby cells with lots of possibilities. Okay, we do have these things called apical meristems that are found at the tips of the roots and the buds of the shoots, so the top and the bottom. The two apices, they're found at the top and the bottom to help continue growth in each of those directions. Those are called apical meristems. Primary growth occurs at the apical meristem, so it allows the roots to push downward and the shoots to push upward. So that's simple as it is. That's primary growth, is up and down. Let me keep moving there, but up and down at these apices or apexes. Those at either end will give us primary growth. So this is primary growth. Okay. And so there are these cool things that root tips are covered by a root cap. We won't really go into the different zones. So, all right, moving on. All right, we're not gonna worry too much about it. But the apical meristem, here's another example. This is the part where we have lots of undifferentiated cells. This is also, we didn't get to look at it, but if we could, this is where you do an onion root, and you can look at it and you can see those cells in constant division. So there's a lot more mitosis, just to give you a kind of a connection to that stuff. It's that there's a lot of mitosis that's happening in that area as they grow. Secondary growth is an increase in the thickness of the stems or the roots. So think thickness is secondary growth is really like, if here's the stem, here, there. It's really just making this thicker. So now we're at this point growing outward. So secondary growth is really kind of just growth laterally or growth at the sides, growth like just getting thicker. Okay, so sideways. There are some lateral meristems. There's the vascular cambium and the corp cambium, but I'm not super worried about getting to know those. There's some secondary xylem and phloem for wood and bark. We're good if we don't know those right this second, but the whole point that I want you to get out of it is that if you do notice how this gets thicker over time, that's secondary growth because we're getting growth in a sideways direction. So if nothing else we get from it, that's our secondary growth. Okay, that we're getting wider or thicker as the years go on. How we do that doesn't matter. What's cool about this, the only little bit that's worth knowing, is that these wood annual rings show layers of secondary xylem. So they actually are what show us how old they are. Rings occur in areas where new growth starts each year. So that's where you get those. Bark, the secondary phloem and cork, is sloughed off over time. But really, this is when we have some dormant times in temperate regions, they'll actually show us a year after year ring situation. So wood rays consist of a bunch of stuff. We're not going to worry about that one. Or sapwood or heartwood. There's just some interesting things. The whole point I want you to get out of it is that you can see parts of the ring structures, and that will tell you how old it is, and that's based on xylem and phloem and based on secondary growth. That's all we get out of that right now. I'm happy because it can get really technical really quickly, but we want to just know the basics. All right, so here's one that's had quite a bit of secondary growth, so much so that they could make a little tunnel out of it. Isn't that nice? All right, so we'll get into reproduction of flowering plants next time. We'll worry about that in the next lecture, but stop there. You know, if you have any questions, shoot me an email.